Vanakkam. Welcome to this video on classics in neuroscience. In this set of videos, we will be looking at signaling molecules, in particular the first growth factor. In this video, we will be looking at axonal growth cone and the organizer principle. Remember, like all cells in the body, neuronal cells or nervous tissue also arise from the embryo, from the single cell. How do connections happen between neurons? This is a question that has been around for several tens of years, more than 100 years. There are several factors, both neurotropic and neurotrophic, that lead to these kind of connections. What is this? Tropic and neurotropic refers to the direction of the action. Neurotrophic refers to the action that is required for survival action. How are these happening? And the discovery or the confirmation of the nerve growth factor or the NGF. This is also something that we will discuss in this set of videos. So, this story begins from the lab of Wilhelm His. The name of the person is His. His is not the pronoun. In this case, His refers to a person, Wilhelm His, in his lab in Basel. It is a city in Switzerland. Observed neuronal development by microscopic examination, that is, observing how development happens by using a microscope. But you cannot continuously observe the embryo using a microscope. That too in the 1880s, there is no way for you to videograph the activity within a microscope. So, what he did was, he took several ages, he took embryos at several ages. So, these are embryos at different ages. So, let us say this 1, 2, 3, 4, let us call this with different names. These are embryos at different ages, that is different embryos at different ages, and he observed these under the microscope. So, he observed a sequence of events. First is, he observed processes, long processes that go toward the ventral zone, which later came to be called as the axon, and then followed by multiple short processes. These short processes, he called them as dendrites. This axon was further studied by Ramon Cajal, of course the famous Cajal, who called the tip of the axon as a distinct entity, and he gave it the name the axonal growth cone. He suggested that the axonal growth cone is a specialized structure that is more like a battering ram that moves, that keeps proceeding until it reaches a target, and then it makes contacts with its target cells. So, it navigates through lots of cells and fibers and finally establishes contact with its target cells. This he called as the axonal growth cone. This line of study is suggested by Hiss and Ramoni Cajal came to be called as the histological tradition. What is histology? That is dissecting cells or tissues at certain ages, in this case embryos at certain ages, and then studying them under a microscope. This is called, or at least in this case we call this as histology. And based on the observations from the microscope, we can infer the mechanisms from those structures that are stained with specific dyes or specific agents and those that are not stained. Okay. So, from that we can understand what mechanisms are there. Remember, this was the only method that was available in the late 1800s and the early 1900s. Techniques such as electrophysiology were yet to be invented or discovered. Physiological approach to this studies came much later. In the good old days, the only method that was available to study was histology. Of course, this meant that you will have to sacrifice the animals or sacrifice the embryos. Obviously, you will have to cut them and you will have to make slices and then make these observations. So, there is always this disadvantage, but this is what was available at that time. Later, this was followed by the work of Ross Harrison, who practically formed this new field, or what we can call as the new field of the experimental histological approach. Harrison studied at Johns Hopkins and in Berlin. He was one of the early pioneers who was trained in Europe. In the good old days, Europe was the powerhouse in research, in scientific research, in scientific inquiry. Only much later in the 1950s, the United States became the research powerhouse. So, Ross Harrison trained in Berlin and he returned back to America to pursue further studies. So, he performed experiments to address certain misconceptions that still persisted about how axons were formed, because some group of people were still claiming that this formation was due to glial cells accumulating together, something of that sort. So, to address these problems, Harrison performed some ingenious, meaning of that word ingenious means smart, good experiments. What are these experiments? What he did was he dissected really small pieces of neural tissue from the frog embryo and he bathed them in frog lymph. So, essentially put them in a glass dish that contained frog lymph and nothing else, and he observed the growth of axons in this controlled environment. There is nothing else, there is just this isolated axon and the lymph all in a dish. And so, he observed this axonal growth in this controlled environment, and what Harrison observed led to some dramatic changes in the way we studied neuroscience from that point. He observed there is this individual outgrowth of axons without any other cells, in the absence of any other tissue. He observed this dramatic outgrowing axons, and their growth cones continued continued to extend into surrounding lymph, suggesting this idea that axons originated from nerve cells and not from glial cells. So, this provided the final evidence to conclude the idea that axons arose essentially from nerve cells and not from a coalescence of glial cells. So, to disprove this idea that there were glial cells that were coalescing to produce the axon, he came up with this experiment. So, science makes progress by disproving ideas. You do not prove ideas, but rather you provide evidence that support an idea, something to keep in mind, and this led to the great revolution. This was one of the early studies that used study in a glass dish, that set of experiments that later came to be called as the in vitro experiments. A huge amount of studies these days are performed in an in vitro environment. What does it mean? In vitro means a particular tissue or cell has been isolated, a set of cells or some tissue has been isolated from its natural environment and put in a dish that could support its growth or culture, and then you study this culture or growth using microscopy. This sort of experiments have exploded from that point, but Harrison's experiments were among the first in this field that used this sort of in vitro experimentation. We later call this as in vitro perhaps, but this was one of the pioneering experiments in which they showed that this in vitro experiments work. So, Harrison described in his research article the particular experimental method that he used for this study, for studying axon formation. Right. This is from his research article. I will share the research article with all of you. You can check this. He says the method which I have used is, in a word, as follows: small pieces of embryonic tissue taken before the histological differentiation of nerve fibers has begun. That is, before the histological differentiation has begun, you take small pieces of embryonic tissue. They are placed in hanging drops of lymph, and the sealed preparation is kept under observation for a number of days. It is found that the embryonic cells under these conditions manifest striking amoeboid activities, which are especially pronounced in cells taken from the nervous system, and result in such cases in the formation of long threads of hyaline protoplasm. These fibers bear a perfect morphological resemblance to undoubted nerve fiber found in sections of normal embryos of a corresponding stage of development. So striking is the similarity between these structures that no hesitancy is felt in regarding them as identical with one another, essentially suggesting that this method, the in vitro method, can be used to study this sort of development, at least in nerve tissue. So, in the first half of the 20th century, there were not many methods or experimental approaches to study development. The only methods that were available were tissue culture and model systems like eggs of sea urchins, large eggs of animals like sea urchins, and you can separate cells by using ligation. What is ligation? Ligation means you tie different cells using some thread like material. For example, the early ligation techniques used human hair, tying using threads. In this case, the early experiments used human hair. Look how incredibly inventive these scientists were. So, I am having, say for example, 4 cells, that is the 4 cell stage of this embryo. Then I am tying this part and this part. So, I am ligating using human hair, for example. Essentially, I have separated the cells, right, using this tying method, and then you perform one of two things. One is ablation, which is essentially destroying certain cells, some of these cells, or moving one of this cell to another part, or transplantation. Trans means to take from one part, transplantation means to take from one part and putting it in another part. One key question is, initially you are having a set of undifferentiated cells, that is a set of cells that are just cells, all of which look alike, initially at least, that later go on to become specialized cells, that later go on to differentiate themselves. How is this happening? How do you know which set of cells or which cell is going to differentiate into which set of cells or which group of cells? How is this happening, or how is this decided? It is a key question in development. Hans Spemann studied in Würzburg in Germany. Hans Spemann studied in Würzburg in Germany, and he established a school, a place called Freiburg, also in Germany. He was an expert in microsurgical techniques, and he was one of the early pioneers who established that the dorsal part of the egg is essential for organizing the development of the embryo. This is something that he established very early. So, his lab became a training ground for many future developmental biologists. Important students were, one of his important students was Victor Hamburger. Okay. So, in his lab they addressed this question that we posed earlier, which is, how do undifferentiated cells become specialized or differentiated? This was studied by this lady Hilde Mangold. So, Hilde Mangold studied early stages of development, called this stage of development, the cell is called, this is called blastula. Later blastula goes on to form a pore, and then the cells move to the pore and migrate into the interior part of the embryo and form the different organs of the body. This is the early idea of development. What Mangold did was, she took one region of tissue from one cell and transplanted it to a different region in a different developing cell. For example, she took the lip region and transplanted it to the belly region, of the receiving cell, receiving tissue. Okay. So, what she found was that the transplantation led to more neuronal development in the receiving cell, receiving tissue, and sometimes it led to a whole large amount of development. This is something that she found. Sometimes the growth was so much that an entire secondary embryo grew from this. So, based on these, she suggested that the blastospore has some special organizing properties. Now we call as the organizer principle, that this blastopore, this region had some very crucial organizing properties. This organizing properties later came to be called as the organizer principle of development, where she suggested that development is the result of organizer properties of antecedent cell populations. So, remember, this work was performed in collaboration with her advisor Spemann. Unfortunately, Mangold passed away before she received any recognition for this work. Later, this line of work, Spemann was regarded, or he came to be regarded as one of the pioneers of this field. Again, pointing to the fact that would she have received the recognition or any recognition if she was alive? Remember our discussion in the previous week about the discovery of DNA and Franklin, remember that. So, would she have received any recognition if she was alive? Question. So, this is the organizer principle, that is, development is the result of organizer properties of antecedent cell populations. So, the idea that the dorsal lip of the blastopore had special organizing properties, which is called as this organizer principle. This is found in many types of tissues, many different types of tissues to a greater or to a lesser extent. Sometimes this is found even, this is seen even in some foreign substances. But a question is, what organizes the organizer? Where is the information coming to this organizer itself? That is the bigger question. Remember, these experiments were performed only at the level of organs and tissues. These were the good old days, these were the 1930s and 1940s. Similarly, in the field of pharmacology, to identify neurotransmitters, right, same level of experimentation was at the level of organs and tissues. It is just remarkable, it is incredible how much information or how much new knowledge was produced by using these sort of modest or primitive methods. So, once again, going back to our early point about, it is not about the methodology, it is not about how primitive the method is, it is about how well thought out your research question is and how well thought out your experimental approach is. So, with this we come to the end of this video. In this video, we looked at axonal growth cone. In particular, we discussed Harrison's experiment where he allowed growth of nervous tissue in lymph in a dish, in a glass dish, or in vitro experiments, early pioneer of an in vitro experiments. And Mangold's studies on the organizing principles or the organizer principle of the early blastopore, that this region of the dorsal lip of the early blastopore has special organizer principles that later led to the development or to differentiation. Thank you very much for your attention.